Long before the oceans teemed with fish, long before the first shelled creatures carved their place into the sea floor, Earth's waters were home to an alien caste of life. Soft-bodied pioneers, experimental forms, and enigmatic shapes that defied the rules of later ecosystems. In the dim, green-tinged currents of the Ediacaran seas, microbial mats stretched like carpets over the sediment, and upon them sprawled strange, often beautiful organisms, each exploring a different solution to survival in a world without skeletons, jaws, or teeth. Some glided gently across the soft sediment, leaving behind faint, winding tracks. Others stood anchored, frond-like, reaching silently toward the faint sunlight above. Tubular, disc-shaped, or feathery, these creatures experimented with movement, growth, and ecological influence, shaping the seafloor in subtle ways that foreshadowed the complex ecosystems yet to come. This is the story of those early architects of life, the creatures that existed before bones, whose innovations, interactions, and silent mastery of their environment laid the foundation for everything that would follow. The mat makers' microbial mats and the first living landscapes, long before the first soft-bodied animals ventured across the seas, Earth was dominated by life so small it is almost invisible to the naked eye. For billions of years, the planet's oceans were a living carpet of microbes, bacteria, and archaea, forming dense, sticky mats across the seafloor. These microbial mats were far more than static layers of cells. They were bustling ecosystems, complex communities that captured nutrients, trapped sediments, and shaped the very surface of the ocean floor. Stretching for kilometers, they created patterns that would one day fossilize into the earliest evidence of life in the geological record. The mats carried out functions that were astonishing in their complexity. Certain layers of microbes performed photosynthesis, gradually producing oxygen and subtly transforming the chemistry of the oceans and atmosphere. Other microbes processed organic matter, recycling nutrients in a continuous cycle that sustained life across the mats. These living carpets stabilized sediments, formed protective surfaces, and acted as incubators for early multicellular life, creating the first ecosystems capable of supporting more than single-celled organisms. Fossilized stromatolites, layered structures created by microbial mats, provide a window into this invisible world. Some formations rise from the sediment like miniature underwater mountains, their intricate ridges and domes reflecting growth over thousands of years. Others spread flat, forming expansive sheets that ripple like the waves of a silent sea. Each layer of trapped sediment and microbial growth records the passage of time and the activities of countless tiny cells, a testament to persistence without bones, without organs, and without visible structure. Within the folds of these mats, the first multicellular life began to experiment with cooperation. Groups of cells adhered to one another, signaling chemically, dividing labor in rudimentary ways, and creating structures that exceeded the capabilities of any single cell. These early multicellular organisms demonstrated resilience, responding to environmental shifts, optimizing nutrient uptake, and surviving conditions that might have wiped out solitary cells. Their bodies were fragile, yet their coordination allowed them to persist across shifting sea floors. Microbial mats also provided shelter for emerging life forms. Tiny, soft-bodied organisms found refuge within the folds, protected from currents and potential predators. Nutrient gradients inside the mats allowed these early creatures to feed by absorption or surface grazing, long before mouths, digestive tracts, or specialized organs evolved. The mats were not only habitats, but laboratories for life, where evolution could experiment with form, movement, and ecological interaction. The mats interacted with the seafloor in ways that transformed the environment itself. By capturing sediments and altering the chemical composition of the substrate, they shaped the texture of the ocean bottom. Channels, ridges, and small depressions formed naturally over time, creating landscapes that encouraged exploration and settlement by other organisms. This interaction demonstrates that even life without bones could engineer its surroundings, altering the physical world on scales that would influence evolution for millions of years. These communities were dynamic. Damaged sections could repair themselves. New colonies expanded into open spaces, and layers shifted and grew as conditions allowed. Resilience was not built from speed or protection, but from cooperation, adaptability, and continuous renewal. Microbial mats endured chemical fluctuations, temperature shifts, and occasional disturbances, persisting where simpler life might have vanished. The fossil record preserves their legacy in ancient sedimentary rocks, offering one of the earliest glimpses of organized life. The patterns and structures they left behind testify to persistence, complexity, and subtle ecological influence. Life in these mats existed quietly yet profoundly, proving that before bones, shells, or spines, organisms could shape environments, interact with one another, and experiment with forms that would become the foundation for the next wave of life. Dickinsonia, the first gliders. In the shallow seas of the late Ediacaran, over 550 million years ago, a soft, elongated creature drifted across the ocean floor, leaving subtle impressions in the microbial mats beneath it. Dickinsonia was unlike anything alive today, a flat, segmented oval, sometimes stretching more than a meter, with a ribbed, symmetrical body that allowed it to bend, flex, and glide across the seafloor. Its movement was slow and deliberate, a rhythmic undulation that reflected the earliest experiments in coordinated motion among multicellular animals. Fossil impressions revealed Dickinsonia's body in astonishing detail. Each rib-like segment formed a repeating pattern, creating a flexible framework that distributed pressure evenly across its soft tissues. In some fossils, its body appears slightly curved or twisted, suggesting the creature could adjust its shape to navigate uneven surfaces or respond to subtle changes in water currents. Beneath it, faint traces indicate that Dickinsonia, fed by absorbing nutrients from microbial mats, slowly grazing across the carpets of bacteria and algae that dominated the seabed. Its feeding strategy was both simple and revolutionary. Unlike predators that bite or crush, Dickinsonia relied on a slow, direct interaction with its environment, extracting nutrients across its entire ventral surface. This method allowed it to leave a mark on the microbial mats, shaping their growth and altering local nutrient distribution. In effect, Dickinsonia transformed the very seafloor it travelled across, leaving subtle traces that persisted for hundreds of millions of years. The creature's movement was enabled by its segmented body, which may have contained primitive muscular tissues capable of producing undulating contractions. These contractions would ripple along its length, propelling Dickinsonia forward while allowing it to adjust direction with minimal effort. In a world without bones, fins, or rigid supports, this method of locomotion was highly efficient, allowing it to explore the seafloor, seek nutrient-rich areas, and possibly avoid unfavourable conditions. Its body was delicate but resilient. Dickinsonia could withstand minor disturbances, such as shifting sediments or gentle currents, without sustaining damage. Its flat, broad shape allowed it to remain stable on soft surfaces, distributing stress evenly and preventing collapse. In a world where predators capable of tearing or crushing its body had yet to evolve, durability, came from flexibility, not protection. Ecologically, Dickinsonia played a quiet yet important role. By grazing on microbial mats, it influenced nutrient cycling, altered microbial communities, and indirectly shaped habitats for smaller organisms. Its trails may have served as pathways for other creatures, creating rudimentary ecological networks long before the evolution of complex predator-prey interactions. Dickinsonia demonstrates that even soft-bodied animals could impact ecosystems and contribute to environmental complexity. Some evidence suggests Dickinsonia possessed the earliest forms of sensory detection. While lacking a brain or centralized nervous system, it may have responded to chemical gradients, light intensity, or minor disturbances in the water. Such sensitivity would allow it to seek optimal feeding areas and avoid environmental stresses, providing insight into how behavior could evolve in organisms without rigid structures or appendages. The fossil record also hints at remarkable longevity. Dickinsonia existed for millions of years, persisting through environmental changes and fluctuations in microbial mat distribution. Its continued survival demonstrates the effectiveness of soft-bodied design, simple feeding strategies, and ecological adaptability. The creature exemplifies how life could thrive without bones, relying on innovation, interaction, and persistence. Dickinsonia's existence challenges modern perceptions of animal life. Without skeletons, teeth, or claws, it navigated, fed, and interacted with its environment in ways that were foundational for evolution. It was a pioneer, gliding across an alien seafloor, shaping the world around it, and setting the stage for the next wave of multicellular experimentation, a creature entirely defined by flexibility, subtlety, and quiet persistence. Charniodiscus, the frond forests. Above the flat seafloor of the late Ediacaran, anchored firmly in soft sediment, swayed one of the first true architectural organisms of life, Charniodiscus. With a stalk rising from the seabed and a frond-like body stretching into the water column, it created vertical space in an otherwise flat world. Some fronds reached over a metre in length, their delicate branches swaying with the currents, forming underwater forests that provided structure, shelter, and complexity to early marine ecosystems. Charniodiscus absorbed nutrients directly from the water, relying on the constant movement of currents to deliver food to its feathery fronds. Its soft tissues allowed it to bend without breaking, moving rhythmically in response to shifts in water flow. This movement maximized exposure to nutrient-rich currents while minimizing physical stress on its delicate structure. Fossilized impressions capture the intricate branching patterns of its fronds, revealing an organism designed for stability and efficiency in a soft, dynamic environment. The stalks of Charniodiscus were anchored deeply enough to withstand occasional disturbances from sediment shifts, yet flexible enough to bend without snapping. This adaptation allowed the fronds to maintain access to flowing water, even during minor storms or tidal events. By standing tall above the microbial mats, Charniodiscus not only fed effectively, but also created habitats for smaller organisms, forming a rudimentary ecosystem in 3 dimensions. Charniodiscus transformed the seabed around it. By rising above the surface, it intercepted light and water flow, subtly altering currents and shading sections of microbial mats below. Its presence may have facilitated localized nutrient concentration, enabling other early organisms to thrive in areas otherwise dominated by simple microbial layers. These frond forests introduced the first vertical complexity into marine ecosystems, expanding the possibilities for interaction, movement, and survival. The fronds may also have acted as ecological filters. Tiny particles, plankton, or microorganisms could have been trapped or slowed by their surfaces, allowing passive feeding strategies to emerge in smaller organisms. In this way, Chaniodiscus indirectly influenced nutrient distribution and the organization of microbial communities, demonstrating that even stationary, soft-bodied organisms could have significant ecological impact. Its form also hints at early experimentation with symmetry and segmentation. Fossil impressions show a repeating pattern along the fronds, with each segment possibly playing a role in structural support or feeding efficiency. This segmentation foreshadows more complex body plans in later animals, revealing how life gradually explored modular designs that could be adapted for different ecological roles. Chaniodiscus likely reproduced by releasing spores or gametes into the water, spreading its lineage across the seafloor. While direct evidence is scarce, fossil beds containing multiple individuals in close proximity suggest clustering behavior or colonization of favorable habitats. These early communities created the first organized, repeating patterns of life in the oceans, hinting at the foundations of population ecology long before animals with skeletons evolved. Living alongside other Ediacaran organisms, Chaniodiscus occupied a niche that combined stability and exposure. It anchored the seafloor, contributed to habitat complexity, and interacted with currents in ways that shaped the local environment. Its fronds, moving gently in the water, formed a subtle yet persistent presence, demonstrating how soft-bodied organisms could engineer their ecosystems, standing as silent sentinels in the dawn of multicellular life. Chaniodiscus illustrates the power of soft, stationary life to transform a world dominated by flat mats and simple forms. Without bones or rigid support, it thrived, creating space, altering water flow, and influencing the distribution of nutrients and organisms. In the Ediacaran seas, it represents a bold step in ecological innovation, a testament to the ways life experimented with form, function, and interaction long before bones existed. Kimberella, the first grazers. Across the soft, microbial-rich seafloor of the late Ediacaran, a small but remarkable creature moved with purpose. Kimberella, a soft-bodied organism resembling an elongated oval, was one of the first animals capable of active grazing. Its muscular, flexible body allowed it to glide slowly over microbial mats, leaving behind faint traces that would become fossilized for hundreds of millions of years. Unlike stationary frond-like organisms, Kimberella actively interacted with its environment, marking a crucial step in the evolution of mobility and feeding strategies. The most striking feature of Kimberella was its muscular foot, similar in function to that of modern mollusks. This broad, flat surface allowed it to cling lightly to the sediment while pushing forward, scraping, or absorbing nutrients from the microbial mats beneath it. Fossilized traces reveal the distinctive scratch marks it left behind, evidence of feeding behavior that transformed the very surface of the ocean floor. Kimberella was not merely surviving, it was shaping its environment with every glide, creating new textures in the mats, and influencing the growth of other microorganisms. Its soft, flexible body was a marvel of design for a world without bones. Segmented ridges along its length allowed it to bend, twist, and adjust to uneven seafloors, while its ventral surface maintained continuous contact with nutrient-rich layers. This combination of flexibility and adhesion enabled Kimberella to navigate a dynamic environment, moving toward areas where microbial mats were thickest or richest, and retreating from unfavorable conditions. Feeding was Kimberella's primary ecological role, yet it also had indirect effects on other organisms. By grazing on microbial mats, it redistributed nutrients, created open patches, and left behind traces that could be colonized by smaller creatures. Its activity generated ecological niches, laying the groundwork for more complex interactions among early multicellular life. Kimberella demonstrates that even small, soft-bodied organisms could influence the structure and function of entire ecosystems. Evidence suggests that Kimberella may have had a primitive anterior structure capable of detecting chemical signals in its environment. Though lacking a brain, it may have responded to gradients in nutrient density, water chemistry, or microbial activity, allowing it to seek the richest feeding grounds. Such behavior represents one of the earliest forms of targeted movement in animal life, a precursor to more advanced sensory and motor systems that would emerge much later. Kimberella likely reproduced by laying eggs or releasing gametes into the water, spreading across the seafloor and establishing new populations. Fossil beds sometimes reveal multiple individuals in close proximity, hinting at clustering behavior or preference for specific microhabitats. These aggregations not only suggest reproductive strategies, but also hint at the beginnings of population-level interactions and competition among early grazers. Its survival depended on more than mobility and feeding efficiency. Kimberella's soft body was resilient enough to withstand minor sediment shifts and gentle currents, while its muscular foot provided stability on soft, uneven surfaces. In an environment largely devoid of predators, durability came from flexibility and adaptability rather than protection. The creature's design illustrates how life in the Ediacaran could flourish without bones, relying instead on subtle interactions and ecological awareness. Kimberella's presence in the ancient oceans marks a turning point in the history of life. It was a pioneer of grazing, mobility, and environmental interaction, demonstrating that even soft-bodied animals could actively shape their world. Each glide across the microbial mats left an imprint not only in sediment, but in evolutionary history, setting the stage for the diversity of movement, feeding, and ecological roles that would follow. The story of Kimberella is one of quiet innovation. Its soft, flexible body, purposeful movement, and subtle environmental influence show that the earliest animals were not passive observers, but active participants in shaping their ecosystems. In a world without bones, it found ways to survive, thrive, and interact, a small but pivotal step in the long unfolding of animal evolution. Spriggina, the first segmented animals. Across the seafloor of the late Ediacaran, Spriggina emerged as one of the earliest examples of a truly segmented animal. Its elongated, bilaterally symmetrical body was divided into a series of repeating segments, hinting at a body plan that would later become fundamental for arthropods, annelids, and even vertebrates. Unlike Dickinsonia or Kimberella, Spriggina combined flexibility with a defined anterior region, suggesting a rudimentary head capable of directing movement and interacting with the environment in ways that were unprecedented at the time. Fossils reveal Spriggina's segmentation with remarkable clarity. Each body segment may have contained primitive muscular tissue, allowing coordinated movement along its length. This arrangement enabled Spriggina to crawl across soft sediments, adjust its posture, and navigate the subtle contours of the microbial mats. Its anterior region, distinct from the rest of the body, indicates a possible concentration of sensory cells, hinting at the earliest forms of environmental awareness and directional movement. Spriggina's locomotion was slow but purposeful. By flexing its segmented body and coordinating muscular contractions, it could glide over the seafloor with a degree of control unseen in other Ediacaran animals. This mobility allowed it to search for food sources, avoid areas of low nutrient density, and perhaps even explore new microhabitats, giving it a significant ecological advantage in a world dominated by soft-bodied, mostly stationary organisms. The creature's feeding behavior likely involved grazing across microbial mats, scraping or absorbing nutrients from their surfaces. Fossilized trails show evidence of repeated movement patterns, suggesting that Sprigganer could revisit areas rich in resources or maintain a territory of sorts. Its interaction with the environment went beyond survival, influencing microbial distribution and contributing to the earliest forms of ecological engineering on the seafloor. Segmentation also offered structural advantages. In the absence of bones or hard exoskeletons, the division of the body into repeated units allowed flexibility and resilience. Each segment could move independently to some degree, helping Sprigganer absorb the stress of uneven terrain or minor disturbances in sediment. This modularity was an evolutionary innovation, laying the foundation for more complex body plans in animals that would appear millions of years later. Sprigganer's ecological role extended beyond feeding. Its movements across the microbial mats created trails and depressions that could serve as pathways or habitats for smaller organisms. By physically altering the seafloor, it became an indirect architect of microecosystems, influencing the distribution of nutrients and the organization of early communities. These subtle impacts show that even soft-bodied, pre-bone animals could shape their surroundings in ways that echoed through the evolution of life. Evidence from fossil beds suggests that Sprigganer may have lived in loosely organized groups. Multiple individuals are often found in proximity, oriented in similar directions, which may indicate environmental preferences, social behavior, or responses to chemical gradients in the water. Such clustering hints at early forms of population structure, adding another layer of complexity to the Ediacaran ecosystem. Sprigganer's existence demonstrates the power of evolutionary experimentation. It combined segmentation, mobility, and sensory specialization in a body entirely lacking bones. Its adaptations allowed it to explore, feed, and interact with the environment in novel ways, showing that complexity does not require hard parts. Sprigganer represents a pivotal moment in the pre-bone era, a glimpse of how animals began to develop structures and behaviors that would echo throughout the rest of evolutionary history. The creature's delicate but resilient body embodies the principles of early life on Earth: innovation, adaptability, and subtle ecological influence. As Sprigganer moved across the ancient seas, it shaped the microbial landscape, experimented with movement, and hinted at the future possibilities of segmentation, coordination, and directed behavior, marking another key step in the journey of life before bones. Charnia, the vertical frond architects. Rising from the soft, microbial-laden seabed of the late Ediacaran, Charnia stretched upward like a living tapestry of delicate fronds. Unlike most other pre-bone organisms, Charnia was entirely sessile, anchored firmly to the sediment by a holdfast, while its feather-like fronds extended into the water column. Each frond consisted of repeated branches, creating an intricate, symmetrical pattern that maximized exposure to nutrient currents while offering structure and habitat for smaller organisms that moved among its filaments. Fossil impressions of Charnia reveal extraordinary detail, preserving not just the outline of the fronds, but the subtle segmentation along each branch. This modular construction allowed the fronds to sway gracefully with the currents, bending without breaking, and ensuring that feeding surfaces remained optimally exposed. In essence, Charnia's body was a living water filter, capturing dissolved nutrients and microscopic particles from the surrounding seawater with remarkable efficiency. Its vertical structure brought an unprecedented three-dimensional complexity to the seafloor. Where microbial mats once dominated a flat, monotonous landscape, Charnia introduced layers, height, and shade. Small, soft-bodied organisms could navigate among its fronds, finding shelter from currents or creating niches in the spaces between branches. The presence of Charnia transformed the seafloor from a simple microbial carpet into a dynamic ecosystem, demonstrating that even stationary, soft-bodied organisms could engineer their environments. The feeding strategy of Charnia relied entirely on the movement of water. Unlike mobile grazers, it could not reposition itself, so it adapted by maximizing surface area and branching. Each branch may have functioned as a filter, capturing nutrients that floated past, while the repeated segmentation allowed currents to pass through without causing damage. In this way, Charnia thrived by being attuned to its environment, a passive yet highly effective feeder in a world without bones. Charnia's holdfast anchored it securely to the soft sediments while allowing flexibility in the upper fronds. Fossils show that even in clusters, the organisms could coexist without interfering with one another, suggesting a balance between space competition and resource capture. By forming dense frond forests, Charnia not only occupied space, but structured the environment for others, subtly influencing how currents flowed and how microbial communities were distributed. Reproduction in Charnia was likely a combination of spore release and budding, enabling it to colonize nearby areas and establish new frond forests. Fossil beds containing multiple individuals of similar size suggest synchronized growth, perhaps responding to seasonal or environmental cues. These early colonies hint at collective strategies for survival, ensuring that frond structures could persist even as individual organisms succumbed to environmental stresses. The ecological impact of Charnia extended beyond its own survival. By creating vertical space and microhabitats, it encouraged biodiversity in a time when most life forms were simple, flat, and soft-bodied. Its presence may have influenced the evolution of mobility in other creatures, pushing grazers and small predators to navigate around fronds and explore new niches. In this sense, Charnia was more than a passive feeder. It was an ecosystem engineer, shaping evolutionary possibilities long before bones appeared. The repeated, modular construction of Charnia's fronds also illustrates the early experimentation with symmetry and patterning that would become a hallmark of animal evolution. Each frond segment functioned as part of a whole, an efficient balance of flexibility and structural stability. Through this design, Charnia reveals that even organisms without skeletons could achieve complexity, stability, and ecological influence, demonstrating the sophistication of life before bones. Charnia's presence in the Ediacaran seas paints a picture of early oceans alive with structure, movement, and interaction. Its delicate, feathery fronds swayed in invisible currents, creating a quiet but powerful influence on its surroundings. In doing so, it exemplifies how soft-bodied organisms could transform their habitats, engineer ecosystems, and explore new forms of life, standing as a testament to the ingenuity of evolution in a world entirely without bones. Tribrachidium, the radial enigma. Beneath the quiet waves of the Ediacaran oceans, a creature unlike any living today clung to the seafloor. Tribrachidium was a soft-bodied organism with a distinctive triradial symmetry, unlike the bilateral symmetry familiar to most animals. Its rounded, disc-like body featured 3 spiraling arms extending from a central hub, forming a striking pattern that resembled a natural work of geometric art. Anchored to the sediment, Tribrachidium inhabited shallow waters, feeding in ways that remain mysterious yet ingenious. Fossil evidence reveals that Tribrachidium's surface was covered in fine ridges and grooves. Scientists hypothesize that these structures guided water currents across its body, directing suspended nutrients toward its central feeding region. Unlike grazers such as Kimberella, Tribrachidium remained stationary, relying entirely on the movement of water to deliver microscopic food. Its design reflects a sophisticated understanding of flow dynamics, an evolutionary innovation that allowed it to exploit passive feeding in the nutrient-rich seas of its time. The triradial symmetry of Tribrachidium was unique among Ediacaran creatures. Each arm functioned as both a structural and functional unit, distributing the forces of water currents evenly across the body while capturing food. This arrangement created a balance between stability and efficiency, preventing the organism from being overturned or displaced by gentle waves. In a world without bones or rigid support, radial design provided a durable framework that ensured survival through environmental variability. Tribrachidium's lifestyle was intimately tied to the surrounding microbial mats. Positioned atop these mats, it may have absorbed nutrients released by decomposing microorganisms or filtered suspended particles from the water. Fossilized impressions suggest subtle interactions with sediments, hinting that its feeding activity slightly modified the surface beneath it. By shaping its immediate environment, Tribrachidium participated in early ecological engineering, influencing nutrient distribution and microhabitat formation. The organism's stationary nature allowed it to thrive in clusters. Fossil beds often reveal multiple individuals arranged in close proximity, suggesting that Tribrachidium favored certain seafloor microhabitats where water flow and nutrient availability were optimal. These colonies created a patchwork of radial patterns across the sediment, producing a visual and functional landscape that enhanced the diversity of Ediacaran ecosystems. Tribrachidium's feeding may have been supported by subtle, cilia-like structures or internal channels that guided water and nutrients across the arms. While the exact mechanism remains speculative, its morphology demonstrates evolutionary experimentation with fluid dynamics, an early precursor to more complex feeding adaptations seen in later animals. The combination of form, symmetry, and functionality represents a remarkable step in pre-bone innovation. Its ecological role extended beyond nutrient capture. By occupying specific spaces on the sea floor, Tribrachidium influenced the movement of currents at a micro scale, potentially affecting the settlement of other organisms. Its presence may have encouraged certain grazing patterns, guided microbial mat regrowth, or even created sheltered niches for smaller life forms. In this way, it participated in the earliest known examples of ecosystem engineering, despite being entirely soft-bodied. The radial symmetry and stationary lifestyle of Tribrachidium challenge our assumptions about early animal design. It demonstrates that complexity does not require mobility or hard parts, but can emerge from precise arrangement, functional architecture, and environmental integration. In a world devoid of bones, Tribrachidium thrived by mastering flow, stability, and passive feeding, leaving behind a fossil record that continues to puzzle and inspire scientists today. Tribrachidium's enigmatic form reminds us that early life was diverse, inventive, and often unlike anything we see in modern oceans. Anchored yet interactive, passive yet ecologically influential, it exemplifies the subtle ingenuity of pre-bone animals' life, experimenting with new shapes, strategies, and ecological roles in the silent, microbial-laden seas of the Ediacaran. Hallucigenia, the limbed oddity. In the deep, soft sediments of the Cambrian seafloor, a creature emerged that would baffle scientists for over a century. Hallucigenia was a small, elongated animal, barely a few centimeters long, yet its bizarre body plan sets it apart in the history of life. Its delicate, tubular body was supported by 7 to 10 pairs of slender, spiny legs, while a series of longer, defensive spines arched over its back. It moved slowly across the sea floor, a peculiar, almost alien presence among the microbial mats and early multicellular animals of the Cambrian seas. Fossils reveal Hallucigenia's anatomy in exquisite detail. Its paired limbs were soft but muscular, allowing it to crawl across sediment while probing the surface for food. The spines along its dorsal surface were rigid and likely served as protection against early predators, creating a defensive barrier for such a fragile organism. The combination of locomotion and defense highlights an evolutionary balancing act. Hallucigenia needed to move efficiently while avoiding being eaten, all without bones or a skeleton to provide structural support. Hallucigenia's feeding strategy was specialized. It likely grazed on microorganisms, detritus, and possibly microscopic algae, using its front limbs to explore the surface and its tiny, tubular mouth to ingest food. Its presence in certain fossil beds alongside trace fossils suggests that it could traverse specific paths repeatedly, leaving subtle marks on the seafloor. In doing so, Hallucigenia altered the substrate and contributed to nutrient cycling, a small but significant ecological influence in Cambrian ecosystems. Its limbs demonstrate an early experiment in segmented appendages. Each pair functioned in coordination, providing stability, propulsion, and maneuverability across uneven sediment. This modular arrangement foreshadowed the development of jointed limbs in later arthropods, showing that even soft-bodied, spin-covered organisms could explore complex movement strategies. The creature's delicate body required precise balance. Its spines and legs together created a system optimized for survival in a fragile, predator-rich environment. Hallucigenia's spines were more than passive protection. Fossil evidence suggests they may have acted as a deterrent to early predators or competitors, standing tall above the soft tissues beneath. In a world where hard parts were still rare, such defensive innovations were critical. The spines allowed Hallucigenia to occupy ecological niches that might have been inaccessible to completely soft-bodied organisms, illustrating the early arms race between predators and prey. Despite its alien appearance, Hallucigenia was ecologically significant. By grazing and disturbing sediments, it contributed to the restructuring of microbial mats and the redistribution of nutrients. Its movements created microhabitats, providing shelter or opportunities for other small organisms to feed or inhabit the disturbed sediment. The seemingly bizarre morphology of Hallucigenia was, in fact, a series of elegant adaptations, each playing a role in its survival and the maintenance of its ecosystem. Fossilized specimens suggest that Hallucigenia may have reproduced by laying eggs, spreading new individuals across the seafloor and forming localized populations. Its small size, combined with defensive spines and mobile limbs, allowed it to persist in a rapidly evolving environment where predation and competition were increasing. The creature's evolutionary innovations were subtle but effective, demonstrating the power of body design, flexibility, and defensive strategies in the Cambrian. Hallucigenia's place in the tree of life illustrates the creativity of evolution before skeletons became widespread. Its combination of soft limbs, protective spines, and specialized feeding mechanisms shows that even early animals without bones could develop complex strategies for survival, interaction, and ecological influence. The creature challenges our perception of what early animals could be, blending alien form with functional sophistication. The oddity of hallucigenia highlights the experimental nature of early animal evolution. Each spine, limb, and segment reflects adaptation to a world where survival depended on movement, defense, and environmental interaction. In the Cambrian oceans, it moved quietly, feeding and navigating, shaping sediments, and participating in the earliest predator-prey dynamics, a testament to life's ingenuity long before bones would dominate the seas. Opabinia, the five-eyed hunter, among the teeming seas of the Cambrian, one of the most peculiar creatures of all time scuttled along the seafloor. Opabinia. Its soft, elongated body measured around 7 centimeters, yet it carried an astonishing array of adaptations that made it a formidable presence in its ecosystem. 5 bulbous eyes perched atop flexible stalks, scanning the environment from multiple angles, while a long, flexible proboscis extended from its head, tipped with a small claw-like appendage. This combination of sensory and manipulative organs allowed Opabinia to hunt and feed with precision, despite lacking bones or any rigid support. The structure of Opabinia's body is a study in evolutionary experimentation. Its segmented trunk was fringed with lateral flaps that enabled swimming and subtle maneuvering through the water. These flaps acted like soft fins, providing lift and stability while allowing the creature to hover over the sea floor or propel itself forward. The combination of a flexible body and flaps demonstrates an early form of locomotion that bridged crawling and swimming, allowing Opabinia to explore multiple layers of its marine environment. Feeding for Opabinia was both efficient and specialized. Its tubular proboscis could extend, bend, and grasp, delivering small prey directly to its mouth. Fossil evidence suggests it fed on tiny organisms or organic detritus, probing the microbial mats and sediments below. This method of predation shows a remarkable level of coordination and sensory integration for an animal without bones, enabling Opabinia to exploit food sources that simpler grazers could not reach. Opabinia's 5 eyes were an extraordinary evolutionary feature. Arranged along the top of its head, they allowed the creature to detect movement and light from multiple directions simultaneously. This panoramic vision would have been critical for both locating prey and avoiding predators, providing a sensory advantage in a Cambrian world filled with experimental predators and competitors. The eyes highlight the early development of complex sensory systems in soft-bodied animals. Its ecological role was significant despite its small size. By feeding on microbes and detritus while moving across the seafloor, Opabinia disturbed sediments, redistributed nutrients, and influenced microbial community composition. Its movements created microhabitats, shaping the physical and biological landscape in subtle ways. This interplay between predator, prey, and environment illustrates the beginnings of ecological complexity that would later define marine ecosystems. Opabinia's segmented flaps and flexible body also suggest the ability to maneuver in response to environmental changes. Whether adjusting to currents, escaping potential threats, or stabilizing itself over uneven sediment, it relied on a sophisticated integration of movement and sensory input. Such adaptations indicate that even pre-bone animals were capable of remarkable coordination and environmental awareness. Fossil beds show that Opabinia may have existed alongside other Cambrian innovators, such as Hallucigenia and Anomalocaris. Its interactions with these species whether competitive or predatory, provide a glimpse into an ecosystem defined by experimentation, mobility, and ecological interplay. Soft-bodied though it was, Opabinia was an active participant in shaping the Cambrian seafloor, leaving behind both biological and physical traces of its presence. The body plan of Opabinia demonstrates that complexity does not require bones. Its flexible trunk, lateral flaps, specialized proboscis, and multiple eyes represent a sophisticated integration of form and function. It thrived by sensing, manipulating, and navigating its environment, proving that survival in the early seas depended on adaptability, coordination, and evolutionary ingenuity. Opabinia's unusual anatomy continues to fascinate scientists because it challenges conventional ideas about what early animals could be. It combined multiple specialized adaptations in a soft, flexible frame, navigating, feeding, and interacting in a dynamic ecosystem. In doing so, it exemplifies the ingenuity of life before bones, a creature perfectly suited to its Cambrian world, yet unlike anything that survives today. Anomalocaris, the apex predator of the Cambrian. In the turbulent seas of the Cambrian period, one predator ruled above all others, Anomalocaris. Stretching up to a meter in length, this soft-bodied hunter was unlike any modern animal. Its elongated, segmented body ended in a fan-like tail for propulsion, while a pair of large, compound eyes perched on stalks gave it extraordinary vision. At the front, a pair of spiny, grasping appendages extended forward, ready to snatch prey from the seafloor or mid-water. Anomalocaris was a marvel of evolutionary design, a predator perfectly adapted to a world without bones. Its body was covered in flexible, overlapping flaps that ran along its sides, functioning like fins to undulate gracefully through the water. This allowed Anomalocaris to maneuver with precision, hover over potential prey, or accelerate suddenly in a burst of speed. Despite lacking any rigid skeleton, its structure combined flexibility and strength, enabling both stealth and agility qualities that made it the dominant predator of its time. The mouth of Anomalocaris was equally remarkable. A circular, tooth-lined structure could open wide enough to grasp prey and crush it with surprisingly effective force. Fossil evidence suggests it could feed on trilobites and other hard-shelled animals, making it one of the earliest known predators capable of overcoming armored prey. This innovation in feeding marks a turning point in evolution, triggering the first predator-prey arms races and reshaping Cambrian ecosystems. Anomalocaris's large compound eyes were an evolutionary masterpiece. With thousands of lenses, each eye provided a nearly panoramic view of the environment, allowing it to detect even subtle movements in the water. This exceptional vision gave Anomalocaris the ability to hunt with precision, identify potential threats, and navigate its complex marine environment, a sensory sophistication unparalleled among soft-bodied animals of the time. Its appendages at the front of the body were long, flexible, and spiny, perfectly designed for grasping and manipulating prey. They could bend, twist, and fold, allowing Anomalocaris to snatch creatures hiding in sediments or swimming nearby. Together with its mouth and sensory systems, these appendages formed a coordinated hunting mechanism that exemplified functional integration in soft-bodied animals long before bones would evolve. Anomalocaris's presence dramatically influenced Cambrian ecosystems. As an apex predator, it controlled populations of grazers and other small animals, indirectly shaping the distribution of microbial mats and sediment-dwelling organisms. Its hunting behavior introduced new pressures that drove evolutionary innovation, encouraging adaptations such as hard shells, defensive spines, and escape strategies in prey species. In this way, Anomalocaris was both a consumer and a catalyst of ecological change. The creature's streamlined body, flexible flaps, and sensory sophistication allowed it to explore multiple ecological niches. Whether gliding over the seafloor, hovering in midwater, or probing crevices for hidden prey, Anomalocaris combined mobility, sensory perception, and predatory skill in a single, elegant package. Its adaptations illustrate how soft-bodied animals could achieve ecological dominance through design rather than skeletal support. Fossil beds reveal Anomalocaris alongside other Cambrian innovators, forming a dynamic ecosystem of predators, grazers, and filter feeders. Its hunting behavior, locomotion, and environmental influence underscore the complexity of early animal life and the emergence of structured ecosystems. Anomalocaris shows that even before bones, evolution was capable of producing animals with specialized forms, coordinated function, and profound ecological impact. Anomalocaris represents the pinnacle of pre-bone predation, a creature engineered for survival in a challenging, competitive world. Its body, senses, and hunting strategies demonstrate that dominance in the Cambrian seas did not require rigidity or armor. Ingenuity, coordination, and functional integration were enough to shape the course of early animal evolution. Picaia, the first nerve cord. In the quiet currents of the Cambrian seas, a small, eel-like creature glided effortlessly through the water. Picaia was tiny, no more than a few centimeters long, yet it carried one of the most significant innovations in the history of life. The notochord, a flexible rod that would eventually become the backbone of vertebrates. Its slender, segmented body allowed it to undulate smoothly, propelling itself with gentle waves of motion as it navigated the seafloor and open water alike. Fossil impressions of Picaia reveal a delicate, flattened body with fine segments running along its length. These segments contain muscle blocks, or myomeres, which contracted in waves, enabling the creature to swim with controlled, efficient motion. Its dorsal and ventral fin structures provided stability, allowing Picaia to remain upright and maneuver precisely in its aquatic environment. In a world dominated by soft-bodied grazers and predators, Picaia combined flexibility and balance to explore new ecological niches. Its feeding strategy likely involved filtering microscopic particles or grazing on detritus suspended in the water column. Using subtle undulations of its body and fins, Picaia could hover near the seafloor or ascend into the water column to access food. Fossil evidence shows no complex mouthparts, indicating that it relied on passive intake and small-scale filtration. Despite its small size and simplicity, its impact on the ecosystem was profound, as it added mobility and predation avoidance to the growing complexity of Cambrian life. The notochord provided structural support that allowed Picaia to move efficiently while maintaining a streamlined form. Unlike completely soft-bodied animals, it could resist minor deformations, stabilize its body in currents, and maintain precise swimming patterns. This innovation marked a pivotal step toward more complex body plans, laying the foundation for the vertebrate lineage that would dominate future oceans and eventually the land. Picaia's sensory capabilities were subtle but effective. Its head region contained simple sensory structures, likely able to detect vibrations, light intensity, or chemical gradients. These early senses allowed it to avoid predators, locate feeding grounds, and navigate the increasingly competitive Cambrian seas. In combination with its muscular body and notochord, these adaptations gave Picaia the ability to explore environments inaccessible to slower or less coordinated organisms. The creature's ecological role extended beyond its own survival. By moving through sediments and water, it disturbed microbial mats, redistributed nutrients, and indirectly influenced the settlement of other small organisms. Its presence created microhabitats and pathways that other species could exploit, contributing to the increasing ecological complexity of Cambrian communities. Picaia was both participant and architect in its environment, quietly shaping the seafloor with every movement. Fossil beds containing multiple Picaia individuals suggest that they may have travelled in loose groups, possibly following nutrient gradients or responding to environmental cues. This behaviour hints at early forms of population dynamics and spatial organisation, long before the evolution of advanced social behaviours or structured ecosystems. Its repeated success across Cambrian sites underscores its adaptability and the effectiveness of its innovations. Picaia illustrates how subtle adaptations can have monumental consequences. The combination of a notochord, segmented muscles, and sensory structures allowed a small, soft-bodied creature to navigate, survive, and interact with its environment in ways that set the stage for vertebrate evolution. In the Cambrian oceans, Picaia's quiet, undulating movements marked the beginning of a lineage that would eventually give rise to the most complex animals on Earth. Its unassuming form belies its importance. Picaia may have been small, fragile, and soft-bodied, but it carried within it the blueprint for vertebrate life. It swam through a world of microbial mats, grazers, and early predators, subtly altering the seafloor and demonstrating that even pre-bone creatures could leave an enduring legacy. Wiwaxia, the armoured grazer. In the shallow Cambrian seas, a small but remarkable creature crawled slowly across the soft sediment, Wiwaxia. Measuring just a few centimetres in length, it was covered in overlapping scales and rows of sharp spines, forming a protective armour unlike anything that had come before. Wiwaxia combined mobility with defence, grazing across microbial mats while warding off potential predators, a unique strategy in a world dominated by soft-bodied animals. Fossil impressions reveal Wiwaxia's dorsal surface adorned with intricate, overlapping sclerites, small, hardened plates that formed a flexible but protective covering. Interspersed among these plates were long spines, angled to deter attackers from above. This combination of soft tissue and semi-rigid armour provided a balance between protection and flexibility, allowing Wiwaxia to move while maintaining defences against emerging Cambrian predators. Its body was elongated and bilaterally symmetrical, with a ventral foot-like structure that allowed slow, deliberate movement across the sediment. Wiwaxia grazed by scraping or ingesting microbial mats and detritus from the sea floor, using its ventral surface to maintain contact with its food source. Fossilized trails suggest repeated, meandering paths, indicating that it explored its environment methodically, optimizing nutrient intake while leaving distinctive traces of its presence. Wiwaxia's spines were not solely defensive. Their arrangement may have influenced how currents passed over the body, aiding stability and preventing overturning during locomotion. In effect, these spines served multiple purposes, deterring predators, supporting mobility, and creating microhabitats within the Cambrian seafloor. The creature's design demonstrates how pre-bone animals integrated function and form in elegant, survival-driven ways. Ecologically, Wiwaxia was both consumer and engineer. Its grazing redistributed nutrients across microbial mats, disturbed sediments, and left trails that could be colonized by smaller organisms. By shaping its immediate environment, Waiwaxia created a patchwork of resource-rich zones, influencing the settlement and behavior of other early animals. Its impact extended beyond individual survival, contributing to the emerging complexity of Cambrian ecosystems. The organism's sensory structures, though simple, were likely sufficient to detect chemical cues or vibrations in the water. These inputs would guide its movement, allowing it to locate nutrient-rich areas and avoid threats. Even without bones, specialized sensory awareness enabled Waiwaxia to interact dynamically with its environment, a crucial factor in a period of rapidly evolving predation and competition. Reproduction in Waiwaxia likely involved the release of eggs into the water, spreading its lineage across the seafloor. Fossil beds with multiple individuals indicate potential clustering behavior or habitat preference, suggesting that environmental factors strongly influence distribution. These early patterns hint at the beginnings of population dynamics and localized ecological structuring in pre-bone animal communities. Waiwaxia's combination of armor, mobility, and feeding strategy exemplifies the experimental nature of Cambrian evolution. It balanced protection and function, grazing efficiently while avoiding predators, and leaving physical and ecological traces behind. This dual role of consumer and ecosystem influencer highlights the sophistication achievable by soft-bodied pre-bone organisms. Its small, spiny body may seem modest, but Waiwaxia was a pioneer in integrating defense, mobility, and ecological impact. By navigating, feeding, and shaping its environment, it demonstrates that even in the absence of skeletons, animals could achieve complexity, resilience, and subtle ecological engineering in the burgeoning Cambrian seas. Collinsium, the spiny lobopodian. In the twilight waters of the Cambrian, a creature crawled with both elegance and menace. Collinsium. This soft-bodied lobopodian, distant relative of Hallucigenia, combined mobility with a remarkable array of defensive spikes along its dorsal surface. Measuring only a few centimeters long, Collinsium moved deliberately across the seafloor, its numerous limbs grasping microbial mats, while its spines deterred potential predators. In form and function, it epitomized the creative experimentation of early animal life. Fossil impressions show that Collinsium had a segmented body, with each segment bearing pairs of limbs ending in small claws. These limbs allowed precise movement and stability over uneven sediment, enabling it to graze effectively across microbial mats or climb subtle seafloor structures. Its spines, rigid and pointed, protruded from the dorsal surface, providing protection while maintaining flexibility in locomotion. The combination of mobility and armor demonstrates a sophisticated survival strategy long before the evolution of bones. Collinsium's feeding strategy was similar to other lobopodians. It likely grazed on microbial mats, consuming bacteria, algae, and organic detritus. Fossil traces show subtle scratch patterns indicating repeated movement across nutrient-rich areas, suggesting deliberate exploration of the sea floor. By feeding in this manner, Collinsium influenced sediment structure and nutrient distribution, subtly shaping the microbial ecosystem it inhabited. The spines on Collinsium's back were not merely passive defenses. Their orientation likely disrupted attacks from above and provided slight stability when navigating uneven terrain. These multifunctional adaptations illustrate how pre-bone animals integrated defense, locomotion, and environmental interaction in a single body plan, allowing them to thrive in predator-rich Cambrian ecosystems. Its multiple limbs provided precise control over movement suggesting a degree of coordination that allowed Collinsium to react to environmental changes or obstacles in real time. Such sophistication in soft-bodied locomotion was a hallmark of Lobopodians, demonstrating that complex behaviors could emerge without rigid skeletal support. Each limb operated as part of a whole, enabling stability, exploration, and survival in the dynamic Cambrian seafloor. Collinsium also contributed to ecological interactions in subtle ways. Its grazing redistributed nutrients, while its spines and body movement created microhabitats for smaller organisms. By altering the physical and biological landscape, it helped increase habitat complexity, paving the way for further ecological innovation. Even soft-bodied animals without bones could act as ecosystem engineers, shaping the distribution and behavior of other life forms. Reproduction in Collinsium likely involved egg-laying, dispersing young into favorable microhabitats. Fossil clusters suggest that these animals may have inhabited the same nutrient-rich areas, forming loose communities that shared the same resources while maintaining mobility and defense. These population-level interactions hint at early forms of habitat selection and spatial organization in pre-bone ecosystems. Collinsium's combination of spines, segmented limbs, and grazing behavior illustrates the experimental nature of Cambrian life. It shows that even small, soft-bodied organisms could integrate multiple survival strategies simultaneously, achieving resilience and ecological influence without relying on bones or hard skeletons. Its design reflects both creativity and practicality in the evolutionary toolkit of early animals. In the Cambrian seas, Collinsium moved quietly yet purposefully. Its limbs propelled it across microbial mats, its spines offered protection, and its grazing shaped the sediments it traversed. In doing so, it exemplified how pre-bone animals could thrive, interact, and subtly engineer their environment, a testament to the ingenuity of life before skeletons would dominate the oceans. Morella, the Cambrian survivor. Gliding through the shallow Cambrian seas, Morella was a small, shrimp-like creature that combined simplicity with versatility. Measuring just a few centimeters, it possessed a segmented body covered by a flexible exoskeleton, delicate antennae for sensing the environment, and jointed limbs for locomotion and feeding. Though soft-bodied, Morella demonstrated evolutionary innovations that allowed animals to explore mobility, sensory perception, and survival in a predator-rich ocean long before the dominance of skeletons. Fossil evidence reveals Morella's bilateral symmetry and segmented design, providing both flexibility and structural support. Its multiple appendages allowed precise movement across the seafloor, crawling over microbial mats, swimming in short bursts, or grasping tiny particles of organic matter. This adaptability made Morella a generalist, capable of thriving in various microhabitats, from nutrient-rich sediments to open water near the seafloor. Its feeding behavior was likely opportunistic. Morella may have grazed on microbial mats, scavenged detritus, or captured planktonic organisms drifting in the water column. Fossilized burrows and trace marks suggest repeated paths across the seafloor, indicating systematic exploration of feeding grounds. By redistributing sediments and nutrients, Morella contributed to the ecological complexity of the Cambrian environment, despite its small size. The creature's jointed limbs were a marvel of soft-bodied engineering. Each limb functioned in coordination, allowing Morella to maintain stability while navigating uneven terrain, adjust to shifting sediments, and manipulate small food particles with precision. These adaptations provided both mobility and control, demonstrating how early animals could achieve functional sophistication without bones. Morella's antennae served as sensory tools, detecting chemical signals, vibrations, or water movements that indicated food, predators or environmental changes. These early sensory structures allowed it to respond effectively to its surroundings, avoid danger, and exploit feeding opportunities, highlighting the critical role of perception in the survival of pre-bone animals. Its exoskeleton, though flexible and soft, provided subtle protection against small predators. The thin plates absorbed minor impacts and prevented abrasions as Marella moved across rough sediments or through narrow crevices. Unlike the rigid shells of later arthropods, Marella's exoskeleton maintained mobility while offering a delicate form of defense, perfectly balancing vulnerability and survival in a world dominated by innovation and predation. Ecologically, Marella played multiple roles. Its movement across microbial mats redistributed nutrients, while its feeding shaped microbial populations. Trace fossils indicate that it occasionally interacted with other animals, either by competing for resources or avoiding predators. Its presence contributed to micro-scale environmental structuring, demonstrating that even a tiny organism could influence the broader seafloor ecosystem. Reproduction in Marella likely involved external fertilization, with eggs released into the water column. Fossil beds occasionally reveal clusters of individuals, suggesting that Marella may have favored certain feeding grounds or habitats, forming temporary aggregations that allowed for reproductive success while maintaining mobility. These behavioral patterns hint at early forms of habitat selection and localized population dynamics in pre-bone ecosystems. Marella's combination of sensory adaptation, mobility, feeding strategy, and protective structures illustrates the experimental nature of Cambrian evolution. By balancing vulnerability and function, it carved out a niche that allowed it to survive alongside emerging predators and competitors. Its design shows that evolutionary success depended not on size or bones, but on flexibility, perception, and integration with the environment. Even in the soft, shallow seas of the Cambrian, Marella thrived. Its jointed limbs propelled it across the seafloor, its antennae guided it toward food and safety, and its segmented body maintained stability. In a world still learning to experiment with form and function, Marella was both survivor and innovator, a testament to the ingenuity and adaptability of life before bones. Herdia, the Cambrian apex hunter. In the vibrant, predator-rich seas of the Cambrian, Herdia ruled as one of the most formidable hunters of its time. Closely related to Anomalocaris, this soft-bodied predator reached impressive lengths, with a segmented, flexible body and a series of lateral flaps that allowed precise swimming and hovering above the seafloor. Its compound eyes perched on stalks scanned the surrounding waters, while massive, grasping frontal appendages were perfectly adapted for seizing prey. Herdia was a master of motion and perception, embodying the peak of pre-bone predation. Fossils reveal Herdia's segmented exoskeleton, which combined flexibility with a degree of structural support. Unlike modern rigid skeletons, its overlapping body segments allowed it to bend, twist, and propel itself with agility, while the lateral flaps functioned like fins, stabilizing its movements and facilitating both short bursts of speed and long, gliding swims. This design made it a versatile hunter capable of pursuing prey through complex seafloor terrains. Its feeding apparatus was a marvel of Cambrian evolution. The frontal appendages, tipped with spines, could grasp, manipulate, and bring prey to its circular, tooth-lined mouth. Fossil evidence suggests it consumed trilobites, smaller soft-bodied animals, and detritus, making it one of the earliest predators capable of tackling armored prey. Herdia's hunting strategies exemplified evolutionary ingenuity, proving that even soft-bodied animals could dominate the food chain without bones or rigid support. Herdia's sensory systems were equally sophisticated, Its stalked, compound eyes contained thousands of lenses, providing a nearly panoramic view of its environment. These eyes allowed it to detect subtle movements, track potential prey, and avoid threats, demonstrating that vision was already a critical tool in predator-prey dynamics, long before vertebrates appeared. The integration of sensory perception with highly coordinated limbs allowed Herdia to execute precise attacks, a feat that required remarkable neural and muscular coordination. The lateral flaps along Herdia's body enhanced not only propulsion but also maneuverability. By undulating these flaps in waves, it could maintain stability in currents, hover over microbial mats, or dart suddenly to capture prey. These adaptations illustrate how Cambrian predators exploited body flexibility and flow dynamics to dominate their environments, showing that evolutionary innovation extended beyond bones or rigid exoskeletons. Herdia also played a critical ecological role. Its predation influenced the behavior and evolution of other organisms, promoting the development of defensive features such as spines, shells, and burrowing habits. By controlling populations of grazers and smaller predators, it indirectly shaped the distribution of microbial mats and sediments, becoming a keystone species in its ecosystem despite lacking a rigid skeleton. Fossil beds reveal that Herdia coexisted with other apex predators, including Anomalocaris, creating a dynamic and competitive marine environment. Its interactions likely involved both predation and avoidance strategies, pushing other species toward innovation and adaptation. In this way, Herdia exemplifies the first true arms race in animal evolution, highlighting the ecological pressures that drove complexity in the Cambrian seas. The combination of sensory sophistication, flexible body design, and formidable feeding appendages made Herdia a predator capable of dominating its ecosystem. Every movement, from scanning the waters to striking at prey, relied on a coordinated system of muscles, nerves, and specialized structures, demonstrating that soft-bodied organisms could achieve high levels of functional complexity without bones. Herdia's existence underscores a key lesson about pre-bone life. Evolution experimented with form, function, and behavior in ways that were both creative and effective. Its body plan, feeding strategy, and ecological influence reveal that the Cambrian oceans were a theatre of innovation where survival depended on adaptability, coordination, and ingenuity rather than skeletal support. Even as it swam silently through the currents, hunting, feeding, and shaping its environment, Herdia left an indelible mark on the history of life. It was a predator, an ecosystem engineer, and a testament to the evolutionary possibilities of soft-bodied animals, proving that dominance in the oceans could be achieved through design, function, and perception long before bones became the standard of survival. Amplectobelua, the grasping predator. Amid the bustling Cambrian seafloor, Amplectobelua emerged as a specialized predator, blending speed, flexibility, and extraordinary sensory perception. Closely related to Anomalocaris, this creature measured up to 30 centimeters in length, and its body was built for hunting. Lateral flaps along its segmented trunk allowed it to glide through water with precision, while two formidable frontal appendages lined with spines could grasp and manipulate prey. It was a soft-bodied predator that dominated the microbial mats and early benthic communities with subtle but lethal efficiency. Fossils reveal that Amplectobelua possessed a flexible, segmented body covered by thin, overlapping plates. These segments provided both mobility and slight protection, allowing it to bend, twist, and propel itself efficiently. Its flaps functioned like fins, enabling undulating motion through the water, allowing it to hover, dart, or maneuver around obstacles on the seafloor. This sophisticated locomotion made it one of the most agile hunters of the Cambrian, capable of exploring multiple ecological niches without the need for bones. Its grasping frontal appendages were evolutionary marvels. Each appendage could bend at multiple points, capturing prey with spiny tips that held on to struggling animals. Fossils suggest that it could seize trilobites, soft-bodied worms, and other small organisms, feeding effectively across a variety of prey types. This versatility in feeding strategies made Amplectobelua a successful and adaptable predator in the competitive Cambrian ecosystem. Amplectobelua's mouth, circular and ringed with plates, functioned like a crushing tool. Once prey was captured by its spiny limbs, the mouth processed the meal with precision, an adaptation allowing it to handle both soft and lightly armored animals. This combination of grasping appendages and a specialized mouth demonstrates the complexity achievable by pre-bone predators, integrating multiple functional systems into a single body plan. Its sensory system, including stalked compound eyes, provided a nearly panoramic view of the surrounding environment. These eyes allowed Amplectobelua to detect subtle movements and track prey with incredible accuracy, coordinating attacks with precision. The integration of vision, locomotion, and grasping appendages illustrates the sophisticated evolutionary experimentation occurring long before bones or rigid skeletons appeared. The ecological influence of Amplectobelua was profound. By preying on grazers and small soft-bodied organisms, it shaped population dynamics and indirectly influenced microbial mat growth and nutrient distribution. Its predation forced other species to adapt, whether by developing spines, shells, or escape behaviors, illustrating the first predator-prey evolutionary arms race in the Cambrian seas. Amplectobelua's body plan also demonstrates early experimentation with hydrodynamics. Its lateral flaps and flexible trunk allowed subtle adjustments to currents, maintaining balance while hunting. These adaptations highlight the importance of mobility and environmental integration, showing that even soft-bodied organisms could engineer solutions to complex ecological challenges without bones. Fossil evidence suggests that Amplectobelua coexisted with other apex predators, including Anomalocaris and Herdia, contributing to a highly dynamic ecosystem. Its interactions with prey and competitors highlight the intricate balance of Cambrian marine life, where speed, perception, and functional design dictated survival. The combination of grasping limbs, flexible trunk, and sensory precision made Amplectobelua a model of pre-bone predatory innovation. It navigated, hunted, and influenced its environment with elegance and efficiency, proving that soft-bodied creatures could dominate ecosystems long before the evolution of rigid skeletal structures. Amplectobelua reminds us that the Cambrian oceans were a theatre of evolutionary creativity. Every movement, every hunt, and every interaction was a subtle experiment in form and function, demonstrating the remarkable adaptability and ingenuity of life before bones. Orthrosanclus, the armoured grazer. In the shallow Cambrian seas, a small yet extraordinary creature moved with quiet purpose across the microbial mats, Orthrosanclus. Measuring only a few centimeters in length, it was covered with a combination of overlapping sclerites, dorsal spines, and a flexible shell near its head, providing a remarkable balance of defense and mobility. As a soft-bodied grazer, Orthrosanclus illustrates the subtle ingenuity of life before bones, combining protection, feeding efficiency, and ecological interaction in a single, delicate body plan. Fossil evidence reveals that Orthros body was elongated and bilaterally symmetrical, allowing it to navigate the sediment with precision. Its ventral surface likely contained a muscular foot or similar structure, which enabled slow crawling over microbial mats, feeding on bacteria, detritus, and organic particles. Its dorsal armor and spines protected it from predators while maintaining flexibility, demonstrating a careful evolutionary compromise between safety and movement. The arrangement of spines and sclerites along Orthros body may have influenced how currents passed over its surface, enhancing stability while grazing. These spines not only served as deterrents against attacks from above, but also helped the creature maintain balance on uneven substrates. In a world where rigid skeletons were rare, such innovations allowed soft-bodied animals to survive in predator-rich ecosystems. Orthros feeding strategy was methodical. Its ventral surface scraped or absorbed nutrients from microbial mats, while its flexible body allowed it to move efficiently across the seafloor. Fossilized trails indicate repeated paths, suggesting that it explored the same nutrient-rich areas systematically. Through its grazing, Orthros redistributed organic matter, subtly altering the sediment and influencing microbial community structure, making it an ecosystem engineer on a miniature scale. Its sensory structures, though primitive, likely detected chemical gradients, vibrations, or water movement, guiding it toward food and away from threats. Even in the absence of bones, these early sensory adaptations allowed Orthros to respond dynamically to its environment, highlighting the importance of perception for survival in the Cambrian seas. Reproduction likely involved the release of eggs into the water column, dispersing offspring across favorable feeding grounds. Fossil clusters suggest that Orthros may have aggregated in nutrient-rich areas, hinting at early forms of population, organization, and habitat preference. These behaviors demonstrate that even small, soft-bodied organisms could influence ecological patterns beyond individual survival. Orthros' ecological role extended beyond its own feeding. By moving across microbial mats and disturbing sediments, it shaped microhabitats for smaller organisms, influenced nutrient distribution, and contributed to the patchwork complexity of Cambrian benthic ecosystems. Its subtle yet persistent presence helped define the physical and biological landscape of its environment. The combination of armor, mobility, and feeding strategy in Orthros exemplifies the experimental nature of pre-bone life. It shows how soft-bodied organisms could integrate multiple survival functions into a single body, achieving protection, sensory awareness, and ecological influence simultaneously. Even in its small size and fragile form, Orthros thrived. It grazed, moved, and shaped its environment with quiet effectiveness, illustrating that life before bones was not simple or passive. It was innovative, adaptive, and ecologically influential. In the shallow Cambrian seas, Orthros stands as a testament to evolutionary creativity in the earliest complex ecosystems. Milocunmingia, the first fish-like pioneer. In the Cambrian seas, a small yet groundbreaking creature swam with quiet purpose. Milocunmingia. Measuring only a few centimeters long, it was one of the earliest vertebrate-like animals, carrying within its soft, flexible body the first hints of a backbone. Though tiny and fragile, Milocunmingia represents a pivotal moment in evolutionary history, bridging the gap between soft-bodied pre-bone animals and the vertebrates that would one day dominate the oceans and eventually the land. Fossils reveal that Milocunmingia possessed a notochord, a flexible rod running along its body, providing structural support and enabling more efficient swimming. Paired with segmented muscles, this innovation allowed coordinated, undulating movements, giving it the ability to propel itself through the water with controlled precision. In a world dominated by soft-bodied grazers and predators, such mobility offered both an advantage in feeding and a means to escape from threats. Its body was covered in fine, overlapping scales, providing protection without restricting movement. Unlike the rigid armor of armored grazers like Orthrasanklus, Milocunmingia's scales were light and flexible, a perfect compromise between defense and flexibility. These scales may also have reduced drag in water, enhancing swimming efficiency and demonstrating early adaptations to a fully aquatic lifestyle. Milocunmingia's feeding strategy was likely simple yet effective. With a small, tubular mouth, it consumed plankton, microorganisms, and organic particles suspended in the water column. Fossil evidence suggests that it inhabited shallow marine environments, swimming in mid-water or near the seafloor, capturing microscopic prey with subtle precision. Its role as a small filter feeder illustrates the diversification of feeding strategies during the Cambrian explosion. Its sensory systems, though primitive compared to later vertebrates, included structures capable of detecting light and water movement, enabling it to navigate, find food, and avoid predators. Even without a skeleton, these sensory adaptations gave Milocunmingia a significant survival advantage, allowing it to respond to environmental cues with agility and awareness. The notochord's presence marks a major evolutionary milestone. By providing internal support, it allowed for more efficient locomotion, better balance, and the ability to grow longer without structural collapse. This innovation foreshadowed the evolution of true vertebrae and spines, setting the stage for the vertebrate lineage that would eventually dominate marine and terrestrial ecosystems. Milocunmingia likely lived in small groups or loose aggregations, taking advantage of nutrient-rich waters and safety in numbers. Fossil beds show multiple individuals in close proximity, suggesting that habitat selection and localized population patterns were already emerging in these early vertebrate-like animals. Such behaviors demonstrate the growing ecological complexity even among the smallest Cambrian pioneers. Its ecological impact, though subtle, was important. By feeding on plankton and small particles, Milocunmingia helped regulate microbial populations, contributed to nutrient cycling, and served as prey for larger predators. In doing so, it became an integral component of early marine food webs, linking primary producers and apex predators in a complex, interdependent ecosystem. Milocunmingia's combination of a notochord, segmented muscles, protective scales, and sensory structures illustrates the sophisticated experimentation of pre-bone animals transitioning toward vertebrates. It thrived in a predator-rich world, swimming, feeding, and interacting with its environment with quiet effectiveness. Even in its small, fragile form, it represented a monumental step in the evolution of animal life. In the shallow Cambrian waters, Milocunmingia moved with purpose, a tiny pioneer carrying the blueprint of vertebrate life. Its body design, feeding strategy, and environmental interactions demonstrate that the path to complex, skeletal animals was built on innovation, adaptability, and ecological engagement, all achieved in the era before bones would dominate the seas. Odontogryphus, the sliding grazer. Across the Cambrian seafloor, a small, soft-bodied grazer moved with quiet determination, Odontogryphus. Measuring just a few centimeters long, its flattened, oval body allowed it to slide gracefully across microbial mats, scraping and consuming organic particles with a simple, yet effective feeding apparatus. Though fragile and lacking bones, Odontogryphus played a vital role in the early marine ecosystem, turning even the smallest movements into opportunities to shape its environment. Fossil evidence reveals a body covered with soft, flexible tissue, segmented to allow controlled undulation. Its broad, flat shape helped it maintain contact with the seafloor, distributing its weight evenly to glide across sediments without sinking or disturbing delicate microbial structures excessively. This combination of form and function allowed it to exploit feeding grounds efficiently while avoiding detection by predators. Its feeding mechanism, a simple radula-like structure, enabled Odontogryphus to scrape bacteria and detritus from surfaces. While less specialized than the grasping appendages of predators like Anomalocaris, this feeding strategy allowed it to harvest nutrients across vast stretches of the seafloor. Fossilized trails suggest that Odontogryphus traveled systematically, leaving subtle marks on sediments that provide paleontologists with a glimpse into its daily life. Mobility was aided by a ventral foot-like structure, which allowed the creature to propel itself forward in measured, deliberate motions. Unlike swimmers that relied on flaps or fins, Odontogryphus combined creeping locomotion with grazing, balancing energy efficiency and ecological exploration. Its small size and subtle movement reduced predation risk while enabling it to interact with its environment continuously. The sensory systems of Odontogryphus were basic yet effective. Likely capable of detecting chemical gradients and subtle vibrations, it could locate nutrient-rich areas and avoid danger. These early adaptations in perception allowed it to navigate the seafloor with precision, demonstrating that even the simplest pre-bone animals required awareness of their environment to survive. Ecologically, Odontogryphus acted as both consumer and ecosystem engineer. By scraping microbial mats, it redistributed nutrients and created small disturbances that allowed other microorganisms to colonize. Its feeding behavior subtly shaped the seafloor environment, influencing microbial diversity, sediment structure, and the behavior of coexisting animals. Even small, soft-bodied grazers could leave an enduring ecological impact. Its body, plan, and behavior highlight the diversity of evolutionary strategies in pre-bone animals. While predators relied on speed, grasping appendages, and sensory sophistication, Odontogryphus thrived through persistence, subtle environmental manipulation, and efficient exploitation of available food. Its success illustrates that complexity in behavior and ecological function does not require bones. Reproduction likely involved external egg release into the water column, with larvae drifting until settling in nutrient-rich areas. Fossil clusters suggest repeated habitation in specific feeding grounds, hinting at early patterns of habitat preference and population aggregation. These behaviors demonstrate that even seemingly simple animals exhibited ecological awareness and adaptive strategies. Odontogryphus's presence in the Cambrian ecosystem reinforces the idea that life before bones was highly experimental and diverse. It grazed, moved, and interacted with its environment with subtle ingenuity, showing that survival depended not solely on predation or defense, but on efficiency, persistence, and integration with the ecosystem. Sliding silently across the microbial mats, Odontogryphus was a small but influential participant in the Cambrian seas. Its movements, feeding, and environmental impact may have been subtle, but they left an enduring mark on the evolutionary story, a testament to the complexity and adaptability of life, long before bones would dominate the oceans. Vetulicolians, the filter-feeding experimenters. In the Cambrian seas, a group of unusual soft-bodied animals drifted and swam with a quiet purpose. The Vetulicolians. These early pre-bone creatures were unlike any modern animal, with a body divided into two distinct regions, a bulbous front section and a segmented tail that allowed them to filter feed efficiently while maintaining stability in the water column. Vetulicolians demonstrate the experimental nature of pre-bone evolution, combining mobility, feeding specialization, and ecological impact in ways that would influence later chordates. Fossil evidence reveals that the anterior region of Vetulicolians contained openings along the sides, interpreted as gill slits for filter feeding. Water passed through these slits, carrying plankton and organic particles into the mouth, while the segmented tail provided propulsion. This division of function within the body highlights the early experimentation with anatomical specialization, showing that even soft-bodied animals could partition tasks efficiently long before the evolution of bones or rigid internal structures. Their body design allowed them to occupy a unique ecological niche. Vetulicolians could hover in mid-water, filter small particles from the surrounding currents, and avoid competition with benthic grazers like Odontogryphus or armoured grazers like Orthroxanklus. Fossils suggest that they varied in size, from a few centimetres to over 10 centimetres, allowing different species to exploit slightly different resources or habitats, demonstrating an early form of resource partitioning in the Cambrian ecosystem. The segmented tail provided controlled undulation, enabling both hovering and directional swimming. Vetulicolians could drift passively with currents while feeding or actively propel themselves toward plankton-rich areas. This mobility was critical not only for feeding efficiency, but also for predator avoidance, illustrating that pre-bone creatures could combine energy conservation with survival strategies in a competitive ocean environment. Sensory structures in Vetulicolians were rudimentary but effective. Likely capable of detecting water movement and chemical signals, they could adjust their position in response to nutrient flow or predator presence. These adaptations ensured that filter feeding could occur efficiently while minimizing exposure to danger. In doing so, Vetulicolians demonstrate that early animals could integrate perception, movement, and feeding in a coordinated way, even without bones. Their ecological impact extended beyond feeding. By filtering plankton, Vetulicolians helped regulate microbial populations and influence nutrient cycling in midwater ecosystems. They also served as prey for larger Cambrian predators, linking primary producers to apex predators and contributing to the growing complexity of early food webs. Their presence highlights the multiple layers of interaction and influence that even soft-bodied animals could achieve. Fossil evidence indicates that Vetulicolians may have travelled in loose aggregations or schools, exploiting nutrient-rich currents together while avoiding predators. Such behaviours suggest early forms of social or population-level strategies, indicating that even in vertebrate-dominated ecosystems, coordination and environmental awareness were critical survival strategies. Vetulicolians represent one of the first experiments in filter feeding within the chordate lineage. Their combination of specialized body regions, propulsion, and sensory awareness illustrates that soft-bodied pre-bone animals could achieve remarkable ecological sophistication. They demonstrate that evolutionary innovation often involved structural experimentation and functional integration rather than reliance on skeletal support. In the Cambrian seas, Vetulicolians drifted and swam, feeding, influencing nutrient distribution, and linking trophic levels in subtle but significant ways. They were silent engineers of the water column, proof that pre-bone animals could thrive, interact, and innovate in diverse ecological roles, shaping the evolutionary trajectory that would eventually lead to vertebrates and the complex marine ecosystems that followed. Oddities, the last Cambrian experiments. In the late Cambrian seas, the oceans were teeming with creatures that defy modern expectations. Strange, soft-bodied animals continued to experiment with form, function, and survival strategies long before bones would dominate the oceans. Among these final pre-bone oddities were enigmatic species like Bergessia, Lagania, and Parvuncorina, each with adaptations uniquely suited to grazing, predation, or defense, demonstrating that evolution was still in its experimental phase. Bergessia, a tiny arthropod-like creature, combined mobility with protection. Its segmented, soft body ended in a fan-shaped tail that allowed gentle propulsion, while delicate dorsal spines provided a deterrent to small predators. Bergessia moved slowly along the seafloor, feeding on detritus or microorganisms, leaving behind fossilized trails that hint at systematic grazing patterns. Its body demonstrates the intricate balance between vulnerability and survival, a recurring theme in pre-bone ecosystems. Lagania, in contrast, was an active predator, employing flexible frontal appendages to capture soft-bodied prey. Its elongated body allowed swift, undulating movement, while lateral fins stabilized its path through Cambrian waters. Lagania's presence highlights the diversity of predatory strategies emerging among soft-bodied animals, showing that even without bones, creatures could develop specialized hunting adaptations and dominate particular ecological niches. Parvuncorina, a small, shield-like organism, presents yet another form of experimentation. Its flattened, bilaterally symmetrical body featured raised ridges that may have provided protection and stabilization on the seafloor. Fossil impressions suggest a slow, deliberate movement pattern, possibly grazing on microbial mats or filtering particles from the sediment surface. Parvuncorina exemplifies the subtle interplay between morphology and ecology in early animals. Many of these final pre-bone oddities display combinations of traits seen in earlier Cambrian animals. Segmentation for movement, specialized appendages for feeding, sensory adaptations for environmental awareness, and protective structures all recur in varying forms. Evolution was exploring combinations, refining the balance between mobility, defense, and ecological function in ways that would later inform the vertebrate blueprint. The ecological influence of these creatures was significant. By grazing, filtering, and moving through sediments, they redistributed nutrients and altered microhabitats. Predators shaped prey populations, influencing behavior and morphology. Together, these soft-bodied animals created a dynamic, interactive ecosystem where every movement and feeding strategy had consequences for the environment and for other species. Despite their fragility, these animals persisted. Their strategies demonstrate that survival in the Cambrian seas depended on flexibility, sensory awareness, and the ability to manipulate the environment rather than simply size or rigid skeletal support. They thrived by integrating multiple survival strategies into single organisms, setting the stage for the complexity of life that would follow. Fossil evidence also hints at early experimentation with grouping behaviors. Some species may have traveled in loose clusters or occupied shared feeding grounds, allowing more efficient exploitation of resources while minimizing individual risk. These behavioral patterns foreshadow the social and ecological strategies that would become increasingly important in later animal lineages. In the final pre-bone chapter of Cambrian life, Evolution had produced a kaleidoscope of forms: grazers, predators, filter feeders, and armored oddities, each uniquely adapted to their niche. Their ingenuity demonstrates that the absence of bones did not limit ecological sophistication. Rather, it allowed experimentation, leading to the diverse, dynamic, and interdependent ecosystems of the Cambrian seas. As these creatures swam, crawled, and grazed, they left behind traces, impressions, and fossils that provide us with a window into a world before bones. They represent a final flourish of evolutionary experimentation, a testament to the creativity, adaptability, and ecological innovation of life before vertebrates and skeletons would transform the oceans forever.